Hello, I'm Hannah Donnert with the Collaborative on Health and the Environment. Change is bringing you the latest environmental health science through our partnership calls, webinars, science serves, publications, and social media. I would like to welcome everyone to the launch of Che's newest webinar series, Cancer and the Environment, Science and Opportunities for Prevention. Today's webinar is titled Multifactorial Origins of Cancer, Cancer Studies or Case Studies of Environmental Chemicals and Cancer Risks, and a Systems Approach to Prevention. Our moderator today is Karen Wang, Director of the Collaborative on Health and the Environment. We will leave time following the presentation for a brief Q&A session. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentation. After the presentation, our moderator will read out questions for our speakers to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on the webinar right now is muted with the exception of our moderator and our speakers. The webinar is scheduled to last for 70 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Karen.
Thank you, Hannah. Welcome to the launch of our webinar series, Cancer and the Environment, Science and Opportunities for Prevention. We've been planning this 6-part series for over 4 months and are excited to share with you some of the latest research on environmental health risk factors for cancer. It's great that you guys are joining us today for the launch of our series, which will give an overview on the topic, more about that later. I just wanted to let you know what's coming up. The second webinar in the series will be on March 4th. At 10 a.m. Pacific time, and we will hear from 3 speakers on occupational exposures, water pollution, and air pollution and cancer risks. The third webinar will be on heavy metals, EDCs, and pesticides, and will be on April 15th at 10 a.m. Pacific time. The fourth webinar will be on May 20th at 10 a.m. Pacific time, and will cover the important topic of chemical mixtures, as most of these environmental exposures we are talking about are not happening in isolation. The fifth webinar will be on June 3rd at 10 a.m. Pacific time, and we will hear from speakers about environmental health risks and childhood cancers. And the last webinar in this series will be about disparities and will be held on July 22nd at 10 a.m. Pacific time. You can sign up for our newsletter to get reminders of these webinars and others that we are holding, and find out more about these webinars as more details become available on our website, which is healthandenvironment.org. So today's webinar is entitled The Multifactorial Origins of Cancer, Case Studies of Environmental Chemicals and Cancer Risks, and a Systems Approach to Prevention. We have 3 great speakers with us today. I'm going to introduce all 3 of them now. Our first speaker is Dr. Ted Schettler. He is the Science Director of Science and Environmental Health Network. In addition to being a founding member of CHE, Dr. Schettler is an author of several books on environmental health and is actively working with the Healthcare Without Harm Coalition to improve the environmental performance of hospitals and other healthcare institutions. Dr. Barbara Cohn. She is a senior research scientist of the Child Health and Studies at the Public Health Institute in Oakland, California. Her work focuses on studying the impact of environmental exposures in combination with endogenous responses during pregnancy on metabolic health, reproductive health, cardiovascular disease, and cancer risks across 3 generations. And our last speaker is Dr. Polly Hoppin. She's a research professor and program director for environmental health at the Lowell Center for Sustainable Production at the University of Massachusetts, Lowell. She's also co-founder of the Cancer-Free Economy Network, a national partnership across multiple sectors seeking to catalyze a shift from hazardous chemicals to safer alternatives, with a particular focus on communities disproportionately exposed. We are lucky to have all 3 of you guys here today, and we will be starting off with you, Ted. Thank you so much.
Thank you very much, Karen, for your introduction. In the next few minutes, what I hope to do is provide some context for the presentations that will follow me today, but also for presentations that we're going to be hearing throughout the series that Karen just described. Now, according to the National Cancer Institute, there are more than 100 kinds of cancer, and obviously their causes vary. But what we can say that really applies to virtually all of them is by asking the question about what do we mean by the cause of this or that cancer? Because as we know, most, not all, but most cancers actually arise from a collection of multiple factors, not exposure to just single agents. So this introduces the notion of multi-causality, and there are various ways to model this or to think about it. One of the dominant metaphors for describing this multi-causality is the, the causal webs, which is really quite apt because it not only invokes multiple strands coming together, but also implies important interactions among the strands that actually have an impact on the integrity of the causal web. Now, if we look at what kinds of risk factors tend to get emphasized by organizations really around the world, there are a common list that we see repeatedly. Excess tobacco use, for example, diet and nutrition and their influence on cancer risk, obesity, infectious agents, reproductive factors and hormones, ionizing radiation, genetics, excess alcohol use, UV light exposures, and pharmaceuticals. This is a common list of cancer risks, and they're all very important and they deserve attention. But it's also useful to ask about what's missing and why. And you'll notice that this common list of cancer risk factors really tend to focus on individual behaviors, things that are under the control of individuals that if they modify their behavior, they can actually influence their cancer risk. And there's no question that that's true, but if we think about what's missing, I've given here as an example in the bottom of this slide, some other candidates that we want to think about. Environmental and workplace chemicals linked to cancer, for example. Chemicals that are in consumer products that are completely outside of individual control. Non-ionizing radiation, which is getting more and more attention these days with the increasing cell phone use and the deployment of wireless technologies in our homes and throughout our communities. And then there are neighborhood, community, and ecosystem level variables, including contaminants in the air we breathe, the water we drink, the food that we eat, that can influence cancer risk as well. And we note that some communities have far higher exposures than others, and we're going to be hearing more about that later today and throughout the series as well. So this introduces the idea of the eco-social model of cancer causation, where individuals are progressively nested within families, communities, ecosystems, and societies. So we want to sort of think about how do we model this more expanded notion of causation and the multifactorial nature of cancer causation. The sufficient causal pie model is one way to think about that. This is a model in which it's made quite clear that a cause is not a single component, but it's a minimal set of conditions or events that produces the outcome of interest. So we think about component causes that come together to create a minimal set of conditions that lead to the outcome. So what I've illustrated here is just one sufficient causal pie for lung cancer. And I'll just look at some of the wedges within this pie. For example, if one were to put together defective programmed cell death or apoptosis from a tobacco carcinogen with central obesity and its associated inflammation, with a mutated anti-oncogene from radon exposure, for example, and then another mutated gene from a tobacco carcinogen, and an inherited defect in DNA repair, collectively, that then creates sufficient conditions for a case of lung cancer to develop. But the other implication of this model is that you can begin to assemble these wedges of the pie, sometimes very early in life, but until the pie is complete, the clinical disease does not become manifest. So this then gives some idea about how we might think about cancer prevention, even though some of these wedges may be in place. But there are other combinations that are sufficient to result in cancer development over time as well. And so we can begin to think about what are the implications for cancer prevention. I've illustrated here some other things that we might think about with regard to other sufficient causes of lung cancer. And I want to make the point that whereas tobacco, of course, is the most important cause of contributing cause to lung cancer, it's obviously important, but other exposures are also playing a role, and indeed probably playing a more important role as tobacco use declines, because a number of studies are beginning to show that the proportion of people who were never smokers with non-small cell lung cancer has been increasing over the last 15 years or so. So there's clearly something else at play, even for lung cancer, where tobacco is such a leading cause. And I've indicated here some of the candidates for lung cancer risk factors that we often ignore because we're so fixated on the importance of tobacco. They include, for example, criteria air pollutants, diesel exhaust, dozens of hazardous air pollutants, which are present sometimes at fairly high and unsafe levels in some communities. There are other industrial chemicals, metals, asbestos, silica, radon, a big deal in many parts of the country where it's present in the natural geology, and then radiation. These are just examples of things that we want to think about beyond tobacco when we're thinking about lung cancer and opportunities for prevention. Now, several years ago, an expert group got together to begin to look more carefully at whether they could define what are so-called key characteristics of carcinogens. This is applying primarily to chemical carcinogens, and they published their findings in the Environmental Health Perspectives in 2016. That's publicly available. What they did was start with more than 100 known human carcinogens. These are group 1 carcinogens that had been identified by the International Agency for Research on Cancer. And they looked more carefully at the mechanisms by which those chemicals might be interacting with biological systems to influence cancer risk. And they, after their evaluation, they came up with a list of 10 key characteristics, which I've listed here. I'll just point out, generally, it's no surprise that many of them have the capacity to damage genes and DNA and alter DNA repair, resulting in malfunction of genes and frank mutations. Some of them are electrophilic, meaning that they can attract electrons, and by doing that, they can actually cause adducts on DNA and on proteins. Some of them are able to induce epigenetic alterations, which I'll say more about in just a moment. And then others induce oxidative stress, cause inflammation, suppress the immune system, modulate receptor-mediated effects, cause immortalization so that cells that should be programmed to die don't, and then alter cell proliferation and cell death. Now, the point here is not that every carcinogen has all 10 of these, but rather that every carcinogen that's a known carcinogen has one or more. And some have as many as 8, 9, or even 10. I do want to say just a couple of words about epigenetic alterations, because we're going to be hearing more about that today and in future sessions as well. This is a term that applies to chemicals and other agents that can alter gene expression without altering the DNA sequence. They're not causing mutations, but they're modifying the expression of genes. And there are several mechanisms by which they can do this. The most commonly studied is so-called DNA methylation, where a methyl group is attached either to the gene or to its promoter, which tends to have the impact of silencing that gene. So it keeps it from expressing itself. There are a couple of other mechanisms that have been well identified and studied, including histone modification, and then non-coding RNAs can be produced that also cause gene silencing. And you can see, for example, that if you were to silence a DNA repair gene through an epigenetic mechanism, that DNA damage would not be repaired, which would then allow the accumulation of DNA damage or mutations that might lead to cancer. But some chemicals also lead to reduced DNA methylation, which then leads to inappropriate gene expression. And I've just given one example here in rodent studies done by Gail Prinz and her group have shown that low dose exposure to bisphenol A during development leads to reduced methylation and overexpression of certain cancer-related genes in aging prostates. So this is a developmental exposure that then results in overexpression of a cancer-related gene later in life. Dr. Prinz is going to be presenting on one of the later sessions in this webinar series, so we may hear more about that at that time. But I also want to point out that environmental chemicals are not the only exposures that can result in epigenetic effects, but in fact, nutrition, genetics, age, gender, physical activity, stress, and lifestyle can also influence gene expression through epigenetic mechanisms. So the point here, again, is there are multiple factors that are coming together, working through multiple mechanisms that can influence cancer risk. So to close here, I just want to sort of try to sum this up by using this graphic representation of extrinsic factors that collectively and individually are influencing the internal milieu, the biologic milieu, and they include diet and other habits that often get so much attention, but they also include occupational and environmental exposures, as mentioned, our mental and physical health status, which can have an impact on our internal biology, and then the psychosocial and community level variables as well, and we'll be hearing more about that later today. Collectively, they're in constant communication with the internal milieu, influencing the epigenome, nutritional status, inflammatory status, mutational status, and so on, influencing disease risk, beginning early in life, developmentally, and continuing across the life course with then influence on cancer risk and of course on other chronic disease risk as well. So in summary, multiple multi-level chemical and non-chemical stressors combine over the life course that influence cancer risk in individuals and populations. I see this as a quintessential mixture problem. And as Karen mentioned, we're going to be having a session on mixtures later in this series. Beyond the current overwhelming emphasis then on individual behaviors, this perspective really adds programs and policies that also address workplace, school, community, and societal level contributions to risk, and really helps us identify additional opportunities for cancer prevention. So thanks very much, and I'll pass this off to Barbara. Thank you.
Great, thank you so much, Ted. Barbara's pulling up her slides now, but I wanted to remind our guests and our audience, if you wanted to use the Q&A feature on Zoom to start putting in your questions now, we'll be able to get to them at the end of the webinar. Feel free to start asking them now, though, using the Q&A feature. I'm going to pass this on to Barbara, who will take herself off of mute here in a sec when she's all set to go.
Hello, everyone. I'm grateful for being here today to talk to you about this. We're now going to move to the particular to give you an example of the potential for ancestral exposures to be driving some of the cancer trends that we see in the world today. And while we're going to talk about a case study of DDT, because this is a way to give you this, this introduction, I would just like you to know that we are studying other chemicals in this cohort for other purposes and are trying to address mixtures as well. So today, this is what I will talk about, why we have looked at DDT, why we are looking at multi-generational studies, and then I'll do a brief description of the child health and development studies that has made this 3-generation research possible. I'll tell you what we found both for mothers and their daughters. I'll give you some examples of the promise of big data and relate these findings to disparities in health and talk a little bit about some next steps. So if you were alive 1945 to 2020, you are exposed to DDT. Now this pesticide has been banned in most Western countries, but its legacy remains. And this is just a brief introduction to tell you that the can of DDT was composed of the insecticide itself, this is called PPDDT, and a low-level contaminant called OPDDT that is just a different chemical form. And then over time, our bodies and the world and animals turn this into a metabolite called PPDDE very quickly, and it is the most persistent of these compounds. So why 3 generations? Why do we care? Well, we know that there are windows of susceptibility for breast cancer that differ for people depending on their stage of biological development. So there's firstly the mother during a pregnancy. Her breasts are undergoing great changes in order to make it possible to feed her baby once it's born. And we know this is a vulnerable period for the breast based on many epidemiological studies. Then there's the fetus herself, or actually his self, but we're going to talk about breast cancer today, so we'll talk about the female fetus. And she is developing every single organ system she will ever have in her body, including her own ovaries, where the germ cells that will form the 3rd generation exist. This is what we call a very efficient study. By studying the pregnant woman, we understand a hit that happens to the mother during gestation, to the fetus in utero when all of its organ systems are developing, and to this woman's grandchild who will be formed from these eggs. And that's the focus of our 3-generation studies in the CHDS. I like to mention, of course, that these 3 stages of life are not the same. And the things that are happening in the mom's breast are not the same things that are happening in the fetus's breast. And of course, there's no breast here yet at all. So we don't necessarily expect this thing called the environment to have identical associations with risk in all of these generations. So what is the CHDS? Unbelievably, initiated in 1959, we enrolled 20,000 pregnancies by 1967. It was a partnership with the Kaiser Foundation Health Plan in Oakland, California. And these are some of our real kids who have given permission to use their photos in the 1960s. These children are now 50 years old or in their 50s. Unbelievably, we recruited 98% of eligible families. It was a different time and people had volunteer spirit. And we have information on 20,000 pregnancies and 15,000 women. This is a short summary of the kinds of data that we have. We call the initiating families or mothers the F0, and they enrolled in 59 to 67 when they were about 26 years old. And these are their children born 59 to 67 who are now approximately age 50. There have been multiple follow-ups, which I don't have time to talk about today, but we also have most recently done adult female follow-ups around age 50 of these women and of their children who are now an average age of 20, funded by the California Breast Cancer Research Programs. This is just a brief overview of some of the things that we can do because we have been around for 60 years, not me personally. We have interviews that provide prospective risk factors in multiple domains, many of the ones Ted has already mentioned: behavior, socioeconomic, demographics, pregnancy histories, attitudes, and stressors. We have medical records for accurate data transcribed, which is very important. All these things happening well before the chronic diseases that we study occur, so we're not worrying about recall bias or what women remember after they become sick. We have blood samples that enable assays for biomarkers and environmental exposures in multiple generations, the most drawn from the F0 when they were pregnant, the moms, and actually also the dads in the 60s. And we have done examination studies and further life course studies on these F1 and F2, special exams at age 5, 9 to 11, 15 to 17, 30, 44, and 50 in the children, and an exam at age 20 in the grandchildren, and biospecimens at multiple points in time. For this study, we're going to be talking about the residence cancer and deaths that we track using the California Cancer resources and the residency history and death files and national death files that let us understand which of the members of our cohort have been diagnosed with breast cancer. So just to give you an orientation here, if this is 2018 and I know it's 2020, you'll just have to use your imagination. In 2018, the F0 were about an average age of 80, and by the way, most surviving, about 70 to 80% surviving, fathers and mothers. The F1 are an average age of 55 to 60 now, and they are the original children born into the cohort. And the F2, the grandchildren, are an average age of 20. And this little line here is a very interesting one. This is the F3, the fourth generation, some of whom are not quite born yet. And we intend to continue to be able to move into that generation to understand the impact of exposures at critical periods in their great-grandmothers and how that might have influenced them. So the first thing I want to talk about today is how exposure and outcome windows count, and use the example of DDT and mothers' breast cancer. So this study was published in 2007, and it was somewhat surprising, I think, to some people. So what are you looking at here is the age women, the mothers, were first exposed to DDT based upon their age of birth. DDT was broadly introduced into the environment in 1945. So just by the birth date of these moms, by chance, we had a distribution about equal of women who were exposed before they were 4, in some cases in utero. This is the F0, the mothers now. Then this group 4 to 7, this group exposed 8 to 13, and this group exposed over 14 years. On this axis is the proportionate increased risk in breast cancer that the women in this group had if they had high DDT exposure compared to medium DDT exposure compared to the lowest DDT exposure. And you can see, just looking by these purple bars, is the excess risk that's correlated with high DDT measured in pregnancy declines to 0 once you get to women who only exposed after they were 14. And we interpret this as meaning that the breast of women is more vulnerable before puberty. And in fact, this is something that we learned by looking at radiation in Hiroshima and Nagasaki. It should not be surprising, but it has to do with the developmental stages of the tissue at the time of exposure. And some of these risks are very high. And I just want you to particularly look at this one, because the women exposed in infancy had particularly high risk for breast cancer, again, before the age of 50, which is what we're looking at here. So now we decided we needed to extend this study, and this was just recently published in 2019 in the Journal of the National Cancer Institute. So before we had looked at breast cancer in the mothers up to age 50, and now we extended the window to age 50 to 54 for age at diagnosis. These are two different ages for women. 50 to 54 is the perimenopause. Before age 50 is generally premenopausal breast cancer. And this black box is the average risk that we just showed you on the last slide, which is a 5-fold increase in risk for breast cancer correlated with the highest levels of DDT for women who are first exposed to DDT before age 3 and or between 3 and 13. But now we're going to look at this later age at risk, age 50 to 54, and see what happens. So these women who were not exposed before puberty begin to show risk for breast cancer by the time they reach age 50 to 54, which is about 40 years after exposure. And the interesting thing is, as you can see, 2 things happen. This group, which had such high risks if they were first exposed to DDT before age 3, no longer sees risk at age 50 to 54. By age 50, their cancers appear to have occurred. But this group, the 3 to 13 group, these are women first exposed to DDT in infancy or during puberty. By age 50 to 54, they continue to have an increased risk, not just before 50, but into the 50 to 54 year period. So basically, what our study is able to do is very unique, which is to follow a group of women through time and understand that windows of susceptibility and also windows of outcome can change the risk profile just for this one exposure. And this was, if you want more information, you can see this in the manuscript that we have published. So now what about their daughters? So as I said to you before, the women born in the 60s are now reaching the age of breast cancer risk. And so we decided we would look at the first 130 cases or so of the daughter's breast cancer in relation to their in utero exposure, the mother's measured DVT levels when the mother was pregnant with them. And this is what we found. Well, so the highest quartile now of the OP isomer of DVT was correlated with a fourfold increased risk of breast cancer. This is published in the Journal of Clinical Endocrinology and Metabolism. And this was a very interesting and somewhat worrisome finding. I would like to point out that this size of this risk factor is really bigger than what many people see for some of the more standard risk factors for breast cancer that Ted mentioned before earlier, things like reproductive history or timing of pregnancy. And so we're interested in continuing to follow up this finding. So what do we do next? We would really like to use the power of omics to understand mechanisms, possibly find biomarkers, and possibly, because we know everybody's DVT is not going to be measured in their grandmother or their mother. We want to know whether there are distinct marks or ways to understand both who was exposed, but also who's responding to that exposure. Because as Ted said, not everyone does it uniquely. Everyone is unique. So there are multiple biological systems that are potential targets and mediators of exposure effects, as Ted mentioned. The genome is the one we hear most about these days and where most of the money is being spent. The way the genome is transcribed to proteins, and proteins have been also looked at. And then there's the metabolome, which is what is exactly happened or is happening in the body that might be a marker of all these things that came before. And that is the closest thing to phenotype, the thing we actually observe, such as cancer or obesity. And the epigenome is added here, which Ted has thankfully talked about, because I'm going to talk a little bit about today, because it regulates the expression of the genome and may have an important impact on all of these markers below. So here's an example of one of the ways that we have tried to get to understand mechanisms. In this case, we are looking at the methylation of one breast cancer-related gene, but we also looked at two others, that is known to be related to breast cancer risk in families. And we looked at the DNA methylation now in the daughter's blood at age 50, and to see whether the DVT exposure she had in utero, measured in her mother's blood, was related to the methylation of this one particular breast cancer-related gene, which I'm just showing you one because we don't have that much time. And we can see that the highest level of OP DVT exposure was correlated with higher methylation of this gene. We're a long way from understanding what that means and what the biological implications are, but this is proof of concept that from one generation to the next, there was an association of in utero exposure to this orthopara DVT, which was associated also with daughter's breast cancer, with the level of DNA methylation of a breast cancer-related gene that also is linked to timing and onset of puberty, by the way. So now here's another omics domain that I'm going to introduce you to. We've tried to start looking at the metabolome in the mother's pregnancy serum. The metabolome consists of tens of thousands of individual chemicals that are the responses of a human being to all of the things that Ted put on his slide. Stress, environment, your genetics, and your environmental exposures. And in addition, the newest systems can also measure some environmental contaminants as well at the same time. The wonderful benefit of looking at the metabolome is that the tracings and peaks that you get from the mass spectrometry that you use to measure the metabolome can be saved so that one little small amount of serum can yield important information about the mixtures, not only that come from the outside, but the mixtures that come from the inside, or the endogenous responses of a woman to her own exposure. So this is not the detail is not important, but just would like to show you what we got here. These results have been published recently in the Journal of Reproductive Toxicology. But DDT is correlated with a strong amino acid response in the maternal serum. Amino acids are building blocks of protein, and they have been shown in themselves to be involved in tumor aggression and cancer growth. And DDE, the metabolite of DDT, is associated with a strong fatty acid response. Fatty acids are the building blocks of lipids. They're also the breakdown products of lipids, and they're very much involved in the health of the cell. And they are, you may know them best as some lipids in your blood that are measured as markers of coronary heart disease risk, but they also are related to inflammation and a number of the predictors that Ted just talked to you about that are associated with cancer. But notice the primary compound and the metabolite are not associated necessarily with the same responses. So then what do we do about this? Well, this was a human study, and we don't do experiments on humans. So in addition to DDT, there were many other things these people were exposed to, and we didn't measure them all. And so there's always the uncertainty about whether the DDT in utero was, causal of the maternal responses to DDTs. So now we turn to a mouse study that was done in close collaboration with Michelle Lamerrill, who is a toxicologist, an experimental toxicologist at UC Davis. And this was fascinating. She exposed her mice to DDT levels that were similar to those that were in our cohort in utero, in the mother, and analogous to the levels that would be consistent between the mouse and human studies. And when she did this, she found the same metabolomic signal in the mice as in the people. And the reason why this was really important is she is running a controlled experiment where she is able to have a control group who is absolutely not exposed to DDT and not heavily exposed to other set of environmental chemicals. It's not the same kind of mixture that we face in a human study. So this represents an incredible opportunity where there's close linkage for transdisciplinary science to take what we see in humans and decide whether the mechanism is getting close to be causal by also looking in animals in controlled experiments. And by the way, this is also published in Reproductive Toxicology. So now we're going to talk about a definitely more complicated area, if that's possible, which is the attempt to try to find biomarkers. They're both actionable and markers of risk. And we've started this work very slowly, and it is just a start. And this is a paper, first author is Shizao Li from Emory, and he's recently left there, but he's still doing research in collaboration with us. And what he's done is he's looked at the mixed environmental exposures in our pregnancy serum, DDT, the PFAS compounds, and the PCBs. And he's also aggregated some of the thousands of metabolites that he can measure in our pregnancy serum into communities of metabolites that are correlated with each other. This is very exciting because it actually reduces a problem from tens of thousands of chemicals to hundreds. And so one of the things that we see here is really interesting, that DDT and PFAS chemicals, which are per-fluorinated compounds that are in the news now, that were the topic, for example, of the Dark Waters movie, share some of the metabolite features, responses in the mother with DDT. So this opens a really interesting opportunity that what we found here was that there were a small number of metabolic phenotypes that account for a response to a large class of environmental chemicals. You don't have to read the details here, but these boxes show that there are some metabolites that are shared across these blue boxes, which are metabolite communities, that are shared as responses to more than one environmental chemical. This creates the opportunity for biomarkers and may alleviate the problem of trying to measure all of them as exposure, but instead as markers of response. This is just the beginning of this work. And here, in a preliminary analysis of 50 daughters' breast cancer cases, we can see that some of these metabolite communities are significantly correlated in the mother with the subsequent risk of breast cancer in the daughter 50 years later. So we're looking at the mother's response now that correlates with daughter's breast cancer. And red shows metabolites that were somewhat higher in mothers of cases, and blue shows metabolites that were somewhat lower in the mothers of cases. And it's going to take a while to understand all of this. We've only looked at these first 50, and this work is ongoing, but I wanted to show you what might be possible with data like ours. So disparities, switching to this very important topic, I'm just going to give you a quick summary. A number of legacy chemicals like DDT have been linked to adverse health outcomes, not just breast cancer, but other things, obesity, preterm birth, other things. Many were banned in the 70s, and there's been a significant reduction in serum levels. We see that by comparing the serum in our mothers to their daughters just inside our cohort. But despite these reductions, for example, African Americans in our cohort, which are very well represented, about 22% of our cohort is African American, African Americans in the CHDS had excess levels, serum levels, for more of the 24 environmental chemicals that we've measured in both mothers and in the daughter's generation. So this disparity has not disappeared despite the reduction in serum levels from one generation to the next. So this continued disparity suggests that African Americans have sustained a disproportionate body burden and a different body burden that may contribute to poorer health outcomes or differences in health outcomes in African Americans. And like Ted said, this is not a topic that gets lots of play. People have been much more focused on behaviors, and the issue really is here is that neighborhoods and poverty may have a very strong correlation to physical chemicals that come in and out of the body. And we really need to spend more time on this very important topic. I think this series is going to do that. So in summary, DDTs predict breast cancer in mothers depending on both their age at exposure and their age at outcome. In utero, DDTs predicted breast cancer in daughters and correlate to epigenetic changes in their breast cancer-related genes. Environmental chemicals may share important common metabolic pathways within the body. It leads to prevention opportunities to find biomarkers and may even be actionable. And disparities in environmental exposure have occurred in the 1960s, but they are continuing and may play a role in health disparities. So I want to call your attention to this. There's a special issue of reproductive toxicology. It's either going to be called the womb to breast cancer, or it's going to be called the pregnancy exposome in breast cancer. It's due to be released by March of 2020. Many of the papers are already online. And I am a guest editor along with Mary Beth Terry from Columbia University. And I urge you to take a look at it when it comes out. We're very proud of it. The metabolomics papers are there, and some papers on PFASs and daughter's breast cancer are there, plus some other work on breast density that I didn't have time to talk to you about today. What about the grandchildren? I want you to be aware that we're now completing work on DDT during grandmother pregnancy and risk in their granddaughters. Please stay tuned. Those papers are being written now and will be submitted soon. And then there's the question of the future of our cohort. The CHDS, the Child Health and Development Studies, right now is the only opportunity for 3 and upcoming now 4th-generation cancer research on environmental chemicals. We've only begun to understand these risks with the data set. We are actively seeking funds to secure the cohort for the future of science, not necessarily for research, although we take that too, but we need to store the biospecimens, continue to communicate with our CHDS members, and enroll the great-grandchildren formally as they're born and receive their biospecimens. If we don't do this now, it will be 60 years before we could ever do this again. More than $100 million has already been invested in this cohort. If we lose it now, we lose the opportunity to understand windows of susceptibility and ancestral exposures across generations, and to prevent, before they happen, some of the sequelae for environmental and other risk factors that occur way before, even before we were born, and maybe even before our parents were born. So please contact me if you have any ideas or you can help. So we have to thank funders, the California Breast Cancer Research Program that has been wonderfully generous, the National Institutes of Health that has supported the cohort over the years, the state of California, which created and supports the California Cancer Registry, the Vital Status and Department of Motor Vehicle files, without which this cancer birth cohort would not exist. And it really takes a village, and really more like a state and nation. These are only some of the people who have contributed, but I'd like to also point out the CHDS Participant Advisory Council. They are members of our cohort who advise us. They've been doing this for 8 years running. They have become very sophisticated, and they're wonderful and help us greatly. Mary Beth Terry, who has done research on the cohort, Michelle Lamero, a toxicologist who does transdisciplinary work, the people at the EPA, and that is, let's see, go back, because you see them again, Dean Jones at Emory and Giselle Lee at Emory, and many more people that we don't have time to picture here. So anyway, thank you very much.
Great, thank you so much, Dr. Cohn. We'll be pulling up Dr. Hoppin's slides in just a moment here. Again, I wanted to say, if you guys want to put in your questions in our Q&A feature, we'll try to get to as many as we can. We're running behind schedule a bit here, but we will get to as many as we can. And if not now during the webinar, later on we can respond after the webinar. Thanks so much. Dr. Hoppin, if you want to take yourself off of mute, we can hear you.
Great. Can everyone hear me now?
Yes, we can hear you.
Super. Thank you. And thanks so much, Barbara and Ted, for fantastic presentations. I'm delighted to be here to do another setting the stage talk for the cancer series and tell you a little bit about the work that we and many other partners have been involved in for the last 5 years to take a systems approach to cancer prevention. The Lowell Center for Sustainable Production, which is based at the UMass Lowell, one of the 4 campuses of the University of Massachusetts, along with many academic community health professional advocacy partners, share premises, and that is that our systems of production and consumption are not only the root causes of environmental and health problems, but they're also significant contributors to the solutions. There's a professor at the University of Arizona, Dan Sarowitz, that's written about the Lowell Center's work and has called it sustainability solutions science. And that work is made possible in part by our very tight relationship with our sister organization, the Toxics Use Reduction Institute. Our work on environmental contributors to chronic diseases embraces the public health framework that many of you are familiar with, listed there in the middle of the slide, with 4 standard approaches to how you address public health issues. But we add an additional element to that, and that is strategic engagement for systems change. And you'll learn a bit more what I mean about that throughout this talk. But it reflects that people's health, as Ted so well described, is determined by their own biology, but also by the larger ecosystem in which they live. And in the case of environmental contributors to cancer, this system that we're going to describe includes the companies that produce and use toxic chemicals to make our furniture, our clothing, our airplanes, our food. It also encompasses the government that regulates those companies. It encompasses the institutions and the people that care for us when we become ill, and a whole other array of sectors and players in the system. And there are dynamics within that system that keeps it stable, that keeps it producing toxic chemicals and making people sick, along with the many other risk factors that Ted described. And there's also the potential for the dynamics in that system to change so that the system is destabilized and can move to a new place. So the idea of this work is that by bounding or defining the system and studying it, we can gain insights about what keeps it doing what it is doing with regard to environmental risk factors and their impact on health, in this case, asking the questions, what's going right in the system and what's going wrong with regard to reducing environmental carcinogens, and what are promising levers or opportunities for interventions that if we carried them out simultaneously across the system could shift those dynamics so that that the system was better at advancing health and well-being. So the work I'm going to describe today is the work of 5 years where we've applied the notion of systems analysis and thinking to the challenge of reducing environmental contributors to cancer, and with many other partners using that analysis as a foundation for then developing a network of organizations from across the system that are working collaboratively towards a common goal. So in my talk today, I'm gonna tell a few quick stories that illustrate the three categories of environmental exposures that we think about in doing this systems work, and to describe the project of systems analysis and collaborative network development, including identifying some insights that have come from that analysis about 4 areas of work that we think is warranted for collective action by a wide range of organizations. So premises, and you can read these yourselves, but we've started with these premises 5 years ago, and over the course of the project, we've revisited them, we've affirmed them, we've expanded them, and each of them suggests needs and opportunities. But they've provided the rationale for this, what we're calling the Cancer-Free Economy Network, which is now a robust initiative which has over 50 organizations. It's funded by the Garfield Foundation and other funders. And I want to call your attention to one bullet in here, in addition to the systems analysis that we'll talk about, and that is the third one, that cancer-focused constituencies have been largely on the sidelines when it comes to research, practice, and advocacy on environmental chemicals and their contributors to cancer. But that those same constituencies are very powerful potential partners. So a major focus in this network's work is to engage those cancer-focused constituencies. So briefly, 3 kinds of exposures that we think about as we look at the system. The first is legacy exposures, and I'm using the example here of Camp Lejeune that many of you know about, I think in North Carolina, where drinking water on a military base was contaminated with various solvents and volatile organic compounds, which came largely from an off-base dry cleaning business, interestingly, and also supply wells that were contaminated by different kinds of on-base sources, leaking underground storage tanks, spill air, kind of thing. And in public health speak, this is an example of secondary and tertiary prevention, really the really exhausting, frustrating work by epidemiologists and by a group of Marines who now live all over the country to document those exposures and associated cancers. But it ultimately has led to the VA identifying these as compensable illnesses that are related to service. And Dr. Cohn's description of DDT is another example of legacy exposures. A second category that we think about is ongoing emissions to indoor and outdoor air, to water, to soil, to food, exposure in products and workplaces. This slide shows data from the National Air Toxics Assessment that found that emissions from a plant in this community, St. John the Baptist, Louisiana, was causing the highest potential risk of cancer from airborne pollutants of any place in the country. And that risk level was 1,505 per million, 1,505 times higher than the EPA established acceptable risk. But as both Ted and Barbara pointed out, keep in mind that this doesn't take into account exposures that some of the residents may receive from other sources. And of the top 10 census tracts in counties with highest cancer risks due to industrial air pollutants in the country, 6 of these are in St. John the Baptist Parish. And the risk is driven primarily by 3 carcinogens: chloroprene, ethylene oxide, and formaldehyde. And those, as we know, are ingredients in a range of products. And 2 census tracts in this parish have cancer incidence rates that are 25% higher than the state. 3 of them exceed the state cancer incidence rate, which is already high, and there are elevations in a range of particular kinds of cancers. So the prevalence of these risk factors in St. John the Baptist Parish suggests a lot of cancer prevention opportunities. And one of those could be reduction in air emissions. The NADA data show that if just 3 of these chemicals were eliminated, that this parish would drop back to the level of additional cancer risk that is assigned by the EPA as acceptable. And if these 3 particular chemicals were to be eliminated in the 108 other census tracts in the country that exceed the acceptable list threshold, then the risk level from hazardous air pollutants in those census tracts would also drop back to the acceptable designation. There's a lot more to this particular story, including nefarious practices by one of the manufacturers, and also a range of other cancer risk factors, and also issues of access to care and whether people are getting sufficient screening and treatment. The next category that we think about is future exposures. They're current, but they also are ones that we need to think about projecting in the future. And the example here is the unconventional gas development, otherwise known as fracking, that's happening around the country. And this is really an opportunity for primary prevention, thinking in advance about what kind of economic development strategies are going on regionally and nationally, and how might those be interrupted so that we develop economic activity that is beneficial to human health and the health of the planet. I won't go into the details of the risks that fracking poses that are related to cancer, but there is early science that is concerning based on exposure to carcinogens and radioactivity, and initial concerns about childhood leukemias and other cancers potentially, but this is early science. So the next thing I want to share with you is a basic framework for how you go about applying a systems thinking approach to problems that are associated with cancer in the environment. The systems approach has a number of different steps to it, and the first one is to define and diagnose the system. And for this step, over the course of about a year, we had systems analysts who interviewed key informants and worked with the partner organizations to organize multiple convenings of different organizations. We call them actors in systems think. And those included community-based groups that represented people that were disproportionately exposed. They included workers and companies that use toxic chemicals, government agencies, environmental groups, so-called C-suite business leaders, academics, cancer charities, advocacy groups, clinicians, researchers. I don't think I've mentioned them all, but a large number of organizations with different roles in the system. And the analysis also drew on structured discussions, in-person meetings, journal research. And out of that process, there came really stories, lots of stories about how the system functions and what its impacts are, and also maps of systems dynamics. And the process of defining and studying the system is highly iterative. An early task is to identify trends that are relevant to where you might think the system has come from, where it is now, and where it might be going. So I'm just going to give you a couple of examples of these trends, not all of them, but this was a fascinating process to go through collaboratively. The first trend that I'm sure many people on the call are familiar with is that the number of chemicals suspected of contributing to cancer is growing. There's the set classifications by the International Agency for Research on Cancer as known, probably or possibly carcinogenic. That is an exhaustive process for individual chemicals being assessed. Ted referenced the growing understanding of the ways in which chemicals that aren't classified as carcinogens impact the hallmarks of cancer, and Barbara talked about that as well, and that expands the universe of chemicals that may contribute to cancer. And then moreover, what we do know pales by comparison to what we don't. So of 85,000 in that ballpark range of chemicals in commerce, we have substantial information on toxicity for only a small percentage. These next few slides on trends in tobacco exposure and cancer incidence are familiar, so I'll go through them quickly, but important trends to understand. So here you see the steady decline in smoking, and we know that that was followed by declines in rates of cancers that are associated with smoking, a great example of prevention working. But we also know that lung cancers in people who never smoked are increasing, and we also know that other cancers some of which are not smoking related, and some of which the links with environmental exposures are particularly well established in the literature, that those are going up, those incidence rates are going up. Here are trends in childhood cancers, the good news being that we have decreasing mortality among children who get cancer, and that is thanks to cancer research and much better treatment. But we also have the disturbing trend of increased incidence of cancer. So this reiterates that continued investment in cancer treatment is essential, but it also needs to be complemented by investment in primary prevention, because it certainly shouldn't be our ultimate goal to have more and more children getting cancer, although they are living better. There are comorbidities associated with cancer treatment. And that comes up in this other trend, which is a really important one, and that is that the science on the role of environmental contributors in exacerbating problems for people who have cancer or have had cancer, that science is growing. So you see here some references to studies that are looking at exposure to various environmental pollutants, but particularly I'll pick out air pollution that increases morbidity problems as well as issues about prognosis and the likelihood of recurrence. This is really an important slide because it's a bridge for constituencies whose jobs are to support and treat people with cancer. And I noted earlier that these are among the constituencies that are have been largely on the sidelines when it comes to environmental contributors, in particular population level interventions. But increasingly, clinicians are getting questions from their patients about environmental risk factors, and members of cancer organizations are requesting more information about potential links with environmental exposures and steps they can take. So this is a really important area of science and also for working with particular constituencies. The next trend I wanted to mention is that states are leading the way. There's been distressing trends in federal policy, as everyone knows, which many organizations that are listening, I'm sure, are both intimately familiar with and also working on. You know, budget cuts, delays and releases of long-awaited analyses, exodus of top-notch career scientists at EPA, et cetera, et cetera. But we can take some solace in knowing that state legislation has been a real force for change, for stronger protections related to toxic chemicals. There have been, Safer States shows that there have been over 100 chemicals management policies enacted in 35 states. Those are the green ones on this slide, and even more bills than that. And this illustrates the value of a systems lens, I think, that when one place that people are prodding in the system, which is the federal regulatory realm, when that realm is proving not promising, it's important that that work continue, but we also need to pursue other opportunities. If you're not familiar with the Massachusetts Toxic Use Reduction Act and its programs, I really encourage you to take a look. TURA has been a real model for programs elsewhere in the US and around the globe. And the European Chemicals Agency has just released its chemical substitution strategy, and that's been modeled after the Massachusetts program, just as an example. It's simply a reporting and planning requirement, but we did an analysis a few years ago, led by Molly Jacobs, my colleague, that shows that as a result of the TURA law, carcinogen use has declined substantially in Massachusetts. Another important trend is that retailers are taking action to reduce or eliminate chemicals of high concern. And in the interest of time, I won't go into the detail here, but there's been some great work by many advocates and campaigns to encourage these actions by different retailers, which obviously have large amount of leverage in the marketplace when they decide to make a change. So here are the trend summaries, and let you look at them. I've mentioned most of them. We then move on in systems analysis to looking at the dynamics and actually mapping the system. And here you see a range of different organizations that are working to describe the system that they're part of. And I mentioned many of the constituencies before. But just to say that the process of doing the systems analysis yields some really interesting strategic insights, but it is also a way of building relationships among people who are not used to working together. And I'll just lift up a report that will soon be released on childhood cancer prevention, which was a joint effort between the American Sustainable Business Council and many other partners. And it was really an exciting thing to bring those constituencies together to work on a common goal. There's an element of systems analysis which is really interesting and illuminating, and that is looking at the feedback loops, the places in the system that are impeding progress toward a goal of reducing environmental chemical exposure. This is a description of one of the feedback loops. So if you start over here on the left side, you have US chemicals policy not requiring testing before marketing. We have therefore minimal understanding of health impacts. We then therefore have industry weighing in and discrediting the scientific evidence that is brought forward. We have scientific norms that scientists embrace and are trained in, understandably, but their orientation toward needing certainty in conclusions about risk and a culture of skepticism and sometimes being willing to come forward and state a concern in a policy context, that all contributes to a dominant narrative, which is we have either a small problem, it's not particularly important, or we have no proof. I'll mention a little bit more elements of a dominant narrative in a minute. And that leads to the public being confused and then some acceptance of health problems as being inevitable, just part of life. And then we therefore don't have enough pressure on the legislators and the executive branch to pass laws and enforce regulation, which then leads, et cetera. So you see the circular nature of this feedback loop impeding progress. Now here's one that's driving progress in a good direction. So the evidence that we do have on health impacts of chemicals and availability of safer alternatives in particular motivates consumers and cancer advocates and others, gets them excited. Retailers start responding to those consumer requests and begin reducing their use of toxics, which then incentivizes the development of less hazardous toxics products and safer jobs, provides examples and motivation, and then further investment. So the question is, how do you drive the scale of these positive feedback loops and quell the feedback loops that are going in the wrong direction? So you see here that the next step in systems analysis is defining success, including developing indicators and doing some collaborative goal setting. I'm going to just zoom through because I know we're getting late here, but to just show you a slide on collaborative goal setting, which has been used in a variety of contexts. Really interesting to look at the role that setting aspirational and generational goals can play in campaigns that allow you to then backcast and determine how you want to get there. But the Cancer-Free Economy Network here at the bottom, this is the guiding star, which the partners in this initiative have come together to embrace. Within our generation, we'll lift the burden of cancer and other diseases by driving a dramatic and equitable transition from toxic substances in our lives, our communities, and our economy to safe and healthy alternatives for all. So this is a key slide showing you one of the maps that we have developed in this mapping process. This is the shows that at the low level here, the green bubbles are tactical projects that we and the network can have direct impact on. The orange bubbles are ones that those tactical projects can directly influence. The gray bubbles are ones that, though we don't have the capacity to directly impact them, we have confidence that our work at the green and orange level will end up influencing them, and so on. The four primary levers that we've identified in the systems analysis process, which are shown at the orange level, are shift the market, change the story, strengthen capacities and build influence, and expand a prevention agenda. And again, I think I won't go into detail on these just in the matter of time, but maybe one example to give you a sense that many people are aware of in the shift the market realm would be Healthcare Without Harm's excellent work on toxic use reduction in healthcare, where they are working on reducing waste and closing incinerators and healthy food and healthy buildings in ways that are really leveraging significant changes in the market. I want to spend just a moment on change the story, which I mentioned earlier about dominative narratives. You know, this is the current dominative narrative about cancer. Cancer's bad luck. We're making a lot of progress in treating it. It's possible to prevent some cancers by avoiding unhealthy behaviors like smoking and a poor diet. The science linking environmental chemicals and cancer is not very strong. Environmental chemicals are responsible for only a small percentage of cancers, and the environment is cleaner than it used to be. And then you can't have a risk-free society. So does that sound familiar to people? That's, we think, characterizes well the dominant narrative. But how about a change in narrative towards something like this? Hazardous chemicals in our air, water, food, and products are important factors in the development of cancer. Cancer results from a combination of genes and environment, including hazardous exposures. Environmental carcinogens are contributing to rising incidence rates of certain cancers. Investment in treatment and cures is important. We also need to invest in prevention by reducing people's exposures, holding government and industry accountable, supporting innovation in the development of safer chemicals and products. And we can create a robust and thriving economy with these investments. So it's a both and narrative that we've identified as being an important one to promote. So the last thing I want to leave you with here, and there's lots more detail that we'd be very happy to share with anyone in a follow-up setting, is the Cancer-Free Economy Network's structure. So this mapping has led to the formation of a network that is organized into 4 groups: the health science group, the shifting market group, the policy and legal group, and the building power group. And each of these groups really represents different sections of the systems map and different constituencies that that work group is strategically engaging with to pull some of the levers that I described. But we're also working together on cross projects, cross work group projects, and we have a staff now of a strategic director as well as coordinators of each of these work groups, which we call nodes, and some support for communications and fundraising. So I want to encourage you to stay in touch with the Cancer-Free Economy Network. I'm going to give you how you can do that in just a minute. But I want to spend just 1 minute more on examples of a couple of the projects that have come out, because the final step in a systems analysis is to identify interventions and implement them, and then to learn from them and continue to fine-tune your series of interventions designed to shift systems change. So here on the left, the Health Science Network is the one that I'm directly involved in with colleagues, and we provide science interpretation and support to the rest of the network, and then strategic initiatives with cancer-focused organizations and health professionals, as well as regional and place-based initiatives. So here's just a couple of quick examples. The Bladder Cancer Advocacy Network, a cancer-focused organization with which we're working because they were starting to get questions from their members about concerns of environmental exposure. So we've done literature review with them and fact sheets and webinars, most recently a webinar called Bladder Cancer and the Brave, at which the Camp Lejeune example was described by Dick Clapp and another bladder cancer expert also. The second here in the middle, this is a joint statement for health professionals that are calling, that we coordinated and are calling for action on reducing the burden of cancer. And it too has a both and message. This has been led by NC Abbott Witherspoon at the Children's Environmental Health Network. And then finally, the American Association of Cancer Research, the largest cancer research organization in the world, has not had much attention at all to environmental links to cancer. but thanks to work by Margaret Kripke, who was on the President's Cancer Panel, luminary scientist in the realm of cancer research, working with the Cancer-Free Economy Network, the AACR had a standalone meeting at which the 3 people on this webinar all spoke, and others last June, focused on environmental carcinogenesis pathways to prevention. We've also been asked to organize an educational session at the upcoming annual meeting of the AACR. So there are many more project examples to share with you in follow-up. I'll just mention the final one, and that is regional initiatives, in this case, Southwestern Pennsylvania, where we've built a network that brings together many of the same constituencies that I mentioned earlier, and now has 5 work groups, quarterly meetings, and developing its own sets of projects. One of which, for example, is having a number of environmental health nurses attend the Oncology Nursing Society annual meeting coming up in April. Their presentations were accepted, and they're going to then be coming back and doing chapter-based presentations and engagement after that. And finally, policy work. I encourage everybody to think about state cancer prevention and control plans as opportunities for inserting environmental perspectives into cancer prevention. The Pennsylvania plan here on the right is an example. Breast Cancer Prevention Partners and others have done fabulous work on a primary prevention plan for breast cancer in California. The moonshot that Vice President Biden initiated has not had much on environment and has now been closed down, but again, an opportunity for thinking about national initiatives. Where could environmental contributors be inserted? And this one on the left, a National Academy Guiding Cancer Control, which does not have anything on environment. So many opportunities there. Last slide. Here's how to stay connected with the Cancer-Free Economy Network, and also my emails on my front slide. Thanks so much for listening, and we look forward to follow up.
Thank you so much, Polly. We are quite over time, so we're going to just do 1 or 2 questions, and then we're going to have to sign off. Somebody asked about the buy-in from large healthcare systems such as Kaiser and others. Could, Polly or Barbara speak to that?
Sure, and I think Ted can too, because Ted's been very involved with that. Actually, if I can pass the baton to Ted, would you be willing to answer that question?
Very briefly, I think that the healthcare systems that are most engaged in thinking about primary prevention are those like Kaiser, where Kaiser is both an insurer and a provider of healthcare services. So they have many incentives to try to reduce the drivers of of cancer that are showing up in their facilities requiring care. That really, in my view, is an outlier. Many healthcare systems are not thinking about what they can do to reduce the drivers of cancer. They're so concerned with providing care and all of the issues around that. Now, I don't want to sweep everybody up in that, but there are systems in healthcare without harm that are doing more like Kaiser, but unfortunately there's a long way to go.
The other thing to say is that there are providers, and we've been starting to work on this. We did a project called Rethink Plastics. We have been with the Plastic Pollution Coalition, which I know is a collaborator also of the Cancer-Free Economy Network. The providers in the Kaiser system have expressed considerable interest in trying to answer the questions their patients are coming up with. And I do think that the work Polly is doing is going to move the system to the point where they will have to address this because they want to serve their patients. And so some of this is just basic education. GYNs and pediatricians are being given some directives by ACOG and also the American Academy of Pediatrics about the potential for environmental contributions to the health of these populations. But the providers themselves have not had the kind of education and access to answer questions comfortably in the face of uncertainties that will always exist about the information about the environment and health. But I think there's progress is being made, but the questioner is right. You're not getting what you need. We know that. And there are many of us working to make it change.
Okay, great. We are over by 10 minutes, so I think that that's all the time we have. Thank you. Thank you to our 3 speakers, and Hannah, we'll hand it back to you.
Great, thank you so much, Karen. We're approaching the end of today's webinar. A video recording will be available on SHEA's website soon, and tomorrow you'll receive an email containing a link to the video. SHEA's next partnership call will take place February 26th and is titled Holding Corporations Accountable for Contamination, the Role of Litigation, Regulation, and Research to Address the PFAS Problem. To learn more and to RSVP, please visit our website at healthandenvironment.org. If you are new to SHEA and would like to stay updated about upcoming events or more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website at healthandenvironment.org. Additionally, if you appreciate these SHEA partnership webinars bringing you the latest environmental health research for free, we encourage you to support SHEA's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank our speakers, Dr. Schettler, Dr. Cohn, and Dr. Hoppen, for taking time today to present, and to you, Karen, for your excellent moderation. Thank you so much for joining us, and have a great day.